Huntington's disease, there's a big push therapeutically to target the Huntingtin protein and try and lower it. Huntingtin protein being the cause of what goes wrong in all cases of Huntington's disease. So for those kind of trials, we need ways to actually measure that those drugs are doing what they're supposed to do. And to do this, we can use things like the immunoassays, which can bind to Huntingtin in the cerebrospinal fluid of patients. And that's been really important so far to show target engagement for different therapies. But it doesn't tell us about where in the brain these drugs may be targeting and if they're lowering Huntingtin across the whole brain or in certain regions. And as all of these different drug therapies have different kind of approaches and different targets in terms of they could be intrathecal ASOs or they could be gene therapies that target the center of the midbrain. That's a huge unmet need, I think, in the field. And one of the reasons why I invited Aline Delva to talk about give an update on the mutant Huntingtin-PET MRI study called mHTT. This is one of the first radio tracers that have been targeting the mutant Huntingtin protein in the brain. So it's exciting to see where that is in terms of clinical development. But that, I think, is a huge unmet need for Huntingtin lowering programs. One thing that didn't come up during the session was, or my session at least, was the ability to look at somatic instability in patients in their life. So somatic instability is this idea that the Huntingtin gene mutation, which is a CAG repeat expansion, although everybody has a allele that they get measured when they get the genetic test, what researchers have found in different tissues in the body, and in particular tissues that are very impacted in Huntington's disease, like the striatum, there are much higher repeats. And it links to some of the findings in the genetic genome-wide association studies, which have shown hits in genes that are involved in DNA maintenance and repair, as well as new findings that have shown that if the actual sequence of the CAG polyglutamine stretch in the Huntingtin gene is slightly different, where you can have CAA insertions or CAGs, but it's still polyglutamine that can have a huge impact on age of onset. So there's a story kind of coming out from all of these different findings that this might be really important in disease progression, but we currently can't measure this in patients over time because we can't really biopsy the brain and the tissue where these the larger expansions are happening. So if we could find a method as a biomarker to show to measure that would be hugely important for the future of upcoming trials and new gene candidates that are hopefully targeting this.